In this slide set, we will explore the structures and functions of nucleic acids, including DNA, RNA, and their roles within cells. The image in this slide is a photograph of the model of the DNA molecule built by Drs. James Watson and Francis Crick in 1953. This model was used to depict their proposed structure of the DNA double helix. The hypothesized structure was derived from X-ray diffraction data generated by Drs. Maurice Wilkins and Rosalind Franklin. The model was constructed with parts obtained from a machine shop. Drs. Watson and Crick published their proposed DNA structure in the journal Nature in 1953. For their work, Drs. Watson, Crick, and Wilkins were awarded the Nobel Prize in Physiology or Medicine in 1962. The acronym DNA stands for deoxyribonucleic acid. The acronym RNA stands for ribonucleic acid. DNA and RNA are very large biological molecules, also known as macromolecules. DNA and RNA encode the genetic information of all living things. They encode in biological terms the language of life. The elements found in DNA and RNA include carbon, hydrogen, nitrogen, oxygen, and phosphorus. The atoms that make up DNA and RNA are organized into two primary molecular components: bases and pentose sugars with attached phosphate groups. The bases are the key informational components of DNA and RNA. They are the letters of the DNA and RNA alphabet. The bases of DNA include adenine, cytosine, guanine, and thymine, or A, C, G, and T. Each base consists of a nitrogen-containing component called an amine and various side groups specific to the base. Although RNA also uses A, C, and G, RNA does not use T. Instead, RNA substitutes the base uracil, or U, for the T of thymine. The bases of DNA and RNA are attached, as shown in the drawings, to the 1' carbon of a pentose or 5-carbon sugar. This pentose sugar is part of the sugar phosphate backbone of the DNA and RNA macromolecule. Also attached to the pentose sugar is a triphosphate group. The triphosphate group is attached to the 5' carbon of the pentose sugar. Together, the triphosphate group, the pentose sugar, and the base make a molecule called a nucleotide. Nucleotides frequently are used as units of measure to describe the length of a DNA or RNA molecule. For example, a segment of DNA or RNA is often said to be a specific number of nucleotides long. In addition, nucleotides can be used as a measure of distance. In this case, the nucleotide unit describes the number of intervening nucleotides between one position in a DNA molecule and another position in the DNA molecule. For example, one might say that the mutation in gene A that causes disease B is 25 nucleotides away or 25 nucleotides downstream from the first nucleotide of the gene. Note in the drawing on the left how there is only one hydroxyl group attached to the pentose sugar. This hydroxyl group is attached at the 3' carbon position. This makes this pentose sugar a deoxyribose sugar. Deoxyribose sugars are the sugars that make up DNA. In the drawing on the right, there are two hydroxyl groups attached to the pentose sugar at both the 2' and the 3' positions. This makes this pentose sugar a ribose sugar. Ribose sugars are the sugars that make up RNA. DNA and RNA nucleotides join to one another through a single phosphate to form a string or a strand of DNA and RNA nucleotides. When deoxyribose sugar nucleotides join, they make a DNA strand, as shown in the drawing on the left. When ribose sugar nucleotides join, they make an RNA strand. When nucleotides join together to form a strand of DNA or RNA, the terminal two phosphates of the phosphate group are cleaved off, and the remaining phosphate of one nucleotide is joined to the 3' carbon of the last nucleotide in the existing strand. The 5' to 3' orientation of the nucleotides in the diagrams indicate the relative positions of the 5' and 3' carbons of the pentose sugars. Nucleotides are always linked, 3' carbon to 5' carbon, through a phosphate group. DNA strands are synthesized by an enzyme called DNA polymerase. RNA strands are synthesized by enzymes called RNA polymerases. One of the most important features of DNA is that at the physiological pH and temperature of living cells, DNA is usually found as a double-stranded molecule. When DNA forms double strands, the strands are joined together through hydrogen bonds that form between the bases. As shown in the drawing, AT base pairs form through 2 hydrogen bonds, and CG base pairs form through 3 hydrogen bonds. Because of the specificity of this hydrogen bonding, A always pairing with T, C always pairing with G, DNA strands are said to be complementary. The complementarity of base pairing between DNA strands means that knowing the sequence of one strand of a DNA molecule allows one to predict the sequence of the other strand. The length of double-stranded DNA molecules are often described by the number of base pairs in the helix. For example, a DNA molecule containing 27 base pairs of AT and CG from one end to the other would be said to be 27 base pairs long. Base pairs can also be used as a unit of genetic distance. For example, 2 genes can be said to be a specified number of base pairs apart. DNA and RNA strands are oriented according to the ribose sugars within them. The 5' end of a strand of DNA or RNA is the end toward which the 5' carbon of the pentose sugar lies. The strands of double-stranded DNA run anti-parallel or in opposite directions. What this means is that, as shown in the drawing, the 5' end of one strand of the DNA molecule forms hydrogen bonds with the nucleotides at the 3' end of its complementary strand. RNA molecules within cells typically are single-stranded at physiologic temperature and pH. However, they fold upon themselves to form secondary structures wherein a single RNA molecule forms intramolecular base pairs. As with many other biological molecules, the shape created by folding affects how the molecule functions inside cells. Although one RNA molecule may associate with another in some circumstances, RNA does not typically form long, double-stranded intermolecular structures like DNA does. Double-stranded DNA is not a straight ladder-like molecule, however. It turns in a helix like a spiral staircase. The DNA helix is called a double helix because it is made of 2 complementary strands of DNA. The paired bases lie in the center of the spiral staircase, and the sugar-phosphate backbones form the railings of the DNA spiral staircase. The hydrogen bonds that form between the base pairs that hold the sides of the DNA helix together can be broken fairly easily. The DNA double helix can be forced to separate into its component strands by increasing the temperature or the pH of the solution in which the DNA molecule is suspended. If the temperature or pH is lowered back to physiologic levels, the complementary DNA strands will find one another in solution and rehybridize into a DNA double helix, A again pairing with T and C pairing with G along the length of the strands. This physical property of DNA complementarity is the basis not only for replication and transcription of the DNA molecule, but also for most molecular genetic testing technologies through which the sequence of DNA is investigated to diagnose genetic disease, determine paternity, or personal identity. Prior to mitosis, the process by which non-reproductive or somatic cells divide, and meiosis, the process by which germ cell precursors divide to form sperm or egg cells, each DNA double helix within a cell is duplicated. This process, called DNA replication, begins at precise locations encoded within the DNA molecule. These sites are called origins of replication. Enzymes called helicases facilitate the unwinding of the DNA double helix. Once unwound, the DNA strands are replicated by DNA polymerase enzymes. DNA polymerase enzymes copy each strand of the DNA, relying on the complementarity of DNA base pairing to replicate each strand faithfully. Because of the precise process by which each strand of the DNA is used as a template to produce another strand of DNA, DNA replication is said to be semi-conservative. That is, each new DNA molecule is made up of one old strand of DNA and one new strand of DNA. On the leading strand, DNA is replicated in long 5' to 3' segments. However, on the lagging strand, DNA synthesis must proceed 5' to 3' in short fragments called Okazaki fragments. Okazaki fragments are believed to be about 1,000 to 2,000 base pairs in length and are joined together by enzymes called ligases to complete replication of the lagging strand of the DNA molecule. DNA polymerase has a built-in proofreading capability. Because of this proofreading capability, DNA replication is remarkably reliable. In humans, the error rate of DNA replication is believed to be less than 1 base pair per billion. Genes, the instructions for making the RNA and proteins needed to build and maintain cells, are embedded within the nucleotide sequence of the DNA double helix. DNA serves as the repository of these biologic instructions, which consist of a specific series of nucleotides. These DNA instructions are handed down from cell to cell and parent to offspring. In order to implement the instructions encoded within the genes, DNA uses RNA as a messenger. RNA is formed through a process called transcription. During transcription, an enzyme called RNA polymerase utilizes the base pairing between DNA and RNA bases to synthesize an RNA molecule that is a complementary copy of one of the DNA strands. However, instead of incorporating deoxyribonucleotides into the growing strand like DNA polymerase does, RNA polymerase incorporates ribonucleotides into the growing nucleotide strand. The RNA molecule is synthesized in a 5' to 3' direction. Genes have distinct starting and stopping points that are encoded within the DNA molecule. These signals specify where transcription should start and where it should stop. The coding strand of the DNA molecule is the strand that bears the same sequence as the RNA that will be made, with substitution of uracil for thymine. As such, RNA polymerase reads the non-coding strand of the DNA molecule, so that when it synthesizes the complementary molecule, the sequence matches that of the coding strand of the DNA. The coding strand is so called because it is the sequence that specifies the amino acid composition of the protein to be made. The RNA molecules made within eukaryotic cells must be processed before they can be used as messengers of the DNA code. Eukaryotic genes contain introns that must be removed. Introns are non-coding segments of DNA of variable size that separate the coding segments, or exons, of eukaryotic genes. Introns are removed in a process called splicing. In addition to splicing, a methyl group called a 5' cap is attached to the 5' end of the RNA, and a poly-A tail is added to the 3' end of the RNA in a process called polyadenylation. The resulting processed RNA is called a messenger RNA. Processed messenger RNAs migrate from the nucleus, where they are transcribed and modified, to the cytoplasm for translation by ribosomes. Prokaryotic genes do not typically contain introns, although there are some exceptions. As such, there is usually no splicing of RNA molecules in prokaryotic cells. Prokaryotic RNAs are also not capped or polyadenylation as eukaryotic RNAs are. Further, in prokaryotic cells, there is no nucleus to separate the processes of transcription and translation. So transcription and translation in prokaryotic cells often occur simultaneously, with RNA molecules being translated into proteins as soon as they are transcribed from the prokaryotic DNA. A newly discovered branch of the tree of life is the domain archaea. Members of the domain archaea are prokaryotes, just like bacteria. However, some genes of members of the domain archaea have introns and share other structural and functional similarities with eukaryotes. After processing, messenger RNA is transported from the nucleus of eukaryotic cells to the cytoplasm, where it comes into contact with ribosomes. Ribosomes are the protein-making machinery of the cell. Ribosomes progress down the messenger RNA strand in a 5' to 3' direction, creating a protein as they go. Ribosomes are complex structures made up of protein and RNA and can be found floating freely within cells or attached to the rough endoplasmic reticulum. Transfer RNA is the mediator for translation. tRNAs of eukaryotes are transcribed by an enzyme called RNA polymerase III. tRNAs include an anticodon loop, which contains a 3-nucleotide segment of RNA that binds to a complementary 3-nucleotide segment in a messenger RNA called a codon. Transfer RNAs also have an attached amino acid. When a tRNA anticodon loop associates under the guidance of the ribosome with its complementary messenger RNA codon, the amino acid attached to the transfer RNA is moved onto the growing protein molecule. As the ribosome progresses down the messenger RNA strand, additional tRNA molecules come in, bind to the codons embedded within the nucleotide sequence of the RNA, and add amino acids to the growing polypeptide chain, as specified by the nucleotide sequence of the messenger RNA and the gene from which the messenger RNA was transcribed. In RNA, each 3 nucleotide codon specifies a particular amino acid. As such, a messenger RNA has at least 3 times as many nucleotides as the protein it encodes has amino acids. Messenger RNAs also contain non-translated 5' and 3' sequences that typically serve regulatory or other purposes. Note in the diagram on the left how the anticodon loop of the tRNA molecule is complementary to the 3 nucleotide codon of the messenger RNA. This particular transfer RNA carries a methionine, typically the start or initial codon of a eukaryotic protein. The complementarity of codon and anticodon ensures proper amino acid incorporation into growing protein molecules. The table on the right demonstrates the genetic code. The 20 different amino acids are shown in the center of the table, while the nucleotides of the various codons that encode them are shown around the outside edges, with the first position of the triplet codon shown down the left side of the table, the second position across the top, and the third position down the right side of the table. As shown, there are 64 possible codons, but only 20 amino acids. This makes the genetic code redundant. That is, some amino acids are coded for by more than one codon. Note in the table how several different codons all code for the amino acid serine. The redundancy of the genetic code means that not every change or mutation in the nucleotide sequence within a gene will result in a change in the amino acid sequence of a protein. At the end of each messenger RNA is a final codon called a stop codon. This codon tells the ribosome the construction of the protein is complete. The ribosome then falls off the messenger RNA strand, and the newly completed protein is released for further processing and migration to its final intra- or extracellular destination. In this slide set, we have learned that DNA is a complex macromolecule made of several basic atomic components. DNA encodes genes, the instructions needed to make RNA and proteins. DNA is a double helix. The complementary nature of the DNA helix permits its replication and transcription. DNA is transcribed into RNA. Messenger RNA is the template for the production of proteins. Proteins are the molecules that build and comprise structures and perform enzymatic reactions in cells. Transfer RNA enables the transfer of amino acids into the sequence of the growing polypeptide chain. Ribosomal RNA is a component of the protein-producing ribosomes.